Thank you so much for introduction. I should add that I also occupy several positions in Europe and Abu Dhabi now, so it's more complicated. First of all, I want to say that it's a beautiful city and a beautiful conference and beautiful everything. And I want to thank both organizers for inviting and those of you who came for coming. In the next 30 or maybe 40 minutes, I'm going to talk about many things. I'm going to tell you like the concept, then a couple of minor discoveries, a couple of more major ones, and then as a bonus, the story about the drug which has been now approved, it's a new class of therapy actually for autoimmunity. And it's like a challenge, but it is bundled in this whole single story, so I hope I will manage to do it, especially since Encarnita talk helps a lot, because we are going, to continue the same line, essentially. And the story is about, actually, altogether the price that we are paying for the privilege of having adaptive immunity. So it's a great thing to have adaptive immunity for many reasons, but we are paying quite a lot because it is making lots of decisions, and if you are making lots of decisions, sometimes you make mistakes, right? And they accumulate. These errors accumulate in our memory. That's the story for today, essentially. And some disclosures. I'm a co-founder of my laboratories, and we are happy that MyXCR, the software that many, if not most people on the planet analyzing repertoires are now using, and whatever. I'm just happy that that's the result, and some consulting. So we all know, we think that we know to some extent how it all works in adaptive immunity. When it responding to some challenge, we have proliferating clones of T cells and B cells. I will talk about T cells today. I don't mean that B cells are not important, just I'm a T cell guy mainly. And they are proliferating like a hell. From one cell you can have millions of, or even sometimes up to billions of T cells with the same T cell receptor. They are doing the job, eliminating the pathogen, and they go down because, we don't need that much, right, in our life. But we have the memory. We have remaining clones which remember the antigen because they have T cell receptor in this case. And they remember the tissue. Our body is very geographic in this respect. We remember where to go. And I will give one example today how to illustrate it. And they remember the type of response, which is critically challenging because we are not only remembering that here is the antigen, we are remembering what to do with this particular antigen, because depending on what is the type of the problem we are fighting with, we need different instrumentarium of immune system, of course, to fight with the virus pathogens, with bacteria and fungi and so on. And like the key antigen-specific managers of this process are memory and effector helper T cells, which help immune system to understand what to do with this particular antigen in secondary and repeated infections. But initially, when they make the decision which program to choose, Th1 to fight with the virus or intracellular bacteria, Th2 to fight helminth ticks, Th17 to fight bacterial fungi, Tregs to actually antigen-specifically suppress immune response. It's not only about self-antigens, it's about our memory, about what we are going to be tolerant against, microbiota, some environmental antigens, food, whatever. It's all about actually memory of Tregs, which we accumulate from the very first hours and days of after birth and later on through the life. So they need to make this decision correctly, right? To have the right program to choose and to remember what to do. And it's just not that simple because, many things are going on simultaneously. Viral and bacterial infections come together. Or pathogens have different instrumentarium to confuse our immune system. Actually, they can provoke the mistake made by adaptive immunity when they choose the decision. So they're making this decision, this choice, and remembering it in a complicated environment. And sometimes they are doing it wrong, Luckily, it is not about it. The infection comes and one single clone responds. Many clones respond, many naive T cell clones initially respond, and they all grow to the same or different antigens, and they compete. They compete at the clonal level, clone to clone. My TCR is better. No, my TCR is better. I recognize it. And they compete at the level of programs, like Th1 suppress Th2, Th2 suppress Th1, Treg suppress everybody. So that's how it works. So altogether, when the green ones are the good ones, and yellow are suboptimal, and red one is bad, altogether they are still competing and voting. So it's a democratic decision what type of response actually to initiate and remember against this particular pathogen. And then this balance system, it works absolutely majority times correctly, of course. Our immune system evolved for millions of years, it knows how to do it. In the young age. In the old age, it could be a bit more problematic because of many, but at least two reasons. First of all, naive T cells are just not that abundant. They're still diverse, but not that abundant, and in time, probability of meeting with the antigen in a lymph node, it goes down, okay? Not so many naive clones respond for the new challenge. And then they have a burden of previous memory, because there are old guys sitting saying, oh, we know what to do. No, we know what to do here. These cytokines, those cytokines. So they are doing this, making this decision in not that open, clear mind mood, and some noise around. And this may affect. And the memory which is going to arise is it can be different but it can be very long. And so here is what is shown as one guy working in our lab. He was 47, then he became 50. This happens. Yeah? Louder? I can do like this, okay? That's better? Yeah, sorry. Should I repeat the previous part? No, okay. So I hope it was still clear for you. Yeah, so the man working in our lab, he was 47, then became 50, which happens. And you see that each line here is one clone in his peripheral blood, and they're not going to disappear. They are all still there. And they just there probably forever for all his life. So the memory can be extremely long. And this choice of programs they made is also intrinsically, epigenetically imprinted, probably very, very strong. It's not about maybe the tissues, there is more plasticity, but if you look in peripheral blood and you look which helper T cells, which clones have chosen some programs, you see that's extremely stable in time. So what we did here? We sorted, okay, not we, actually David Price and Cardiff super nicely sorted helper T cell populations from peripheral blood of several healthy donors, okay? And we analyzed the repertoires of T cell receptors, that is what we always do. Th7, Th2, Th1, Th117, Tregs, TFH, whatever. And then we relaxed and we did nothing for like 4 years, waiting for the technologies to develop, which is always important. Right? And then the 10x genomics appeared, and we did the single cell profiling for this very same healthy donors 4 years later. And then we just mapped the clones of these Th1, Th2, whatever, 4 years after they were obtained. Yeah, there is we mapped on the UMAP of these single cells, and you see that Th1 they stay in one particular position. They are still Th1 after 4 years. Okay, they didn't exchange programs or whatever. And Th2A remained Th2A, and Tregs remain Tregs. There was a question how they overlap. They don't overlap. Okay, bona fide Treg or bona fide Treg. These are different clones, and this shows that this memory is going to be very stubborn and stable when adaptive immunity made the decision. Yeah? So it remembers what it's going to do with this particular antigen. And it is also very geographic. There was a nice talk a couple of days ago, or yesterday, no, about these mice with fixed TCR beta chain. We also played with the same story with the data flow from Sloan Kettering from Alexander Rudensky lab. It's a long story, I will not describe it in detail, but what is shown here is repertoires of mice Tregs from different lymph nodes from spleen. Yeah? And you see that each dot is one repertoire, different mice, and you see that they cluster really very well together in this multidimensional space, showing that the repertoire of Tregs in this lymph node is different compared to another lymph node and compared to spleen. So the memory of where the clones reside is also very geographic. So they responded to all what to do, how to do, where to do for a long time or forever in our life. And so then the question is, what happens if the response is incorrect? Okay, unlucky. One of the many thousand decisions is not optimal. And the memory is going to be long. This can lead to a very different set of problems. And there are good things about this and bad. The good things is, okay, that's how vaccination works. That's how herd immunity works. And that's why we are grateful for the nature of having adaptive immunity. And also it's about diagnostics. It was a beautiful talk today about antibodies. You can also see snapshots, and Carnita mentioned classification area, arthritis. You can do it, you can do diagnostics, and in some years this should become available. You look in the repertoire, you see the history of vaccinations, potential autoimmune problems, potential cancer. It should be doable, really, it's a nice thing. And the bad thing is, okay, it's about autoimmunity. If it made a mistake to attack, it is reproducing, and it is about inflammaging, associated with many, okay, inflammaging is a vague term, but here I mean very practical, simple, okay, complicated, but clear thing. They reproduce it again and again, and this creates inflammatory niches for cancer. This is about cardiovascular disease with these soft plaques. It is about metabolic disorders. It is about many things, essentially. And what persuades me in the correctness of this concept is this animal that we, that we studied. It lives in Israel. It's called Spalax. It lives quite long for the rodent, about 20 years. No signs of aging, happy, sexy, no inflammation, no cancer the whole life. So this is strange for us, yes. And we digged what is inside the adaptive immunity. And when we, okay, some short video, if it works, no? Okay, saving time. It's nice animal, okay, let's agree. And when you track the repertoire, just diversity of T-cell receptor repertoire in human blood or in mice spleen here, and in Spalax, okay, we have different lifespan, but anyway, for the 2 years of life of mice, diversity goes down. For our 100, okay, ideally, it also goes down, the diversity, because we accumulate clonal expansions, the same that I have shown above, yeah? And they occupy more and more portion of our homeostasis. In this animal, which has no inflammation accumulated with age, no cancer, the diversity is like stable. They are like, looks like they're not accumulating. They probably respond still with expansion, but they don't have this long stubborn memory. And if you look in the transcriptome, and we now confirming it all with single-cell transcriptomics, they do not accumulate large clones. They don't accumulate clonal programs, so some master transcription factors of T cells, if you know them, they're not accumulated. In Spalax, and checkpoints are not accumulated. And actually Tregs, effector Tregs are not accumulated. Why? Because they don't need regulators, because they don't accumulate that many effectors to control. So they don't have this problem. It is still adaptive immunity because, okay, it adapts, it has diversity of receptors, all this stuff, but its memory is not that long, and this could be beneficial in some aspects and conditions, and, okay, evolutionary niches. So if you look at this from the point of just diverse pathological conditions which may happen with us, this point of view explains a lot of, like, majority, I would say, of what could go wrong, actually. In autoimmunity, okay, we are attacking ourselves, hypersensitivity attack, something that we shouldn't attack, cancer, tolerance, okay, usually it is about some inflammation. We do recognize cancer antigens, but we are not going the right way. The immunity has remembered, okay, was persuaded by tumor to make the wrong choice and reproduces the wrong way of doing things. Other situations, and there is also minor errors. Okay, sorry for this minor error. So it is about a weak attack on self, we can call it minor autoimmunity, okay, or some small conflict with microbiota, or, okay, environmental antigen. Here and there we accumulate these small, wrong, not super optimal decisions, and they create new small inflammatory niches, which can then become the niche for the cancer and cause other problems around. So, and that is how we explain inflammaging, if we try to understand what does it mean. And again, from this point, if you look at what we are trying to do in clinics today, all the approach that we are using, most of immunotherapeutic approaches, are about attempt to persuade immune system to change the decision, okay, suppress the consequences of decision made wrong. So we are trying to interfere with this problem that I tried to describe above. All we are doing. And that is what is doing many miracles in clinics right now, because this, okay, anti-inflammatory drugs, checkpoint inhibitors, they suppress like branches of adaptive immunity or vice versa, although the whole immune response to elicit. It is a rude sort of instruments, okay, because they do not operate antigenic specificities. And the clones which are the problem, the clones which made this mistake, the clones which drive the autoimmunity or wrong response to cancer, whatever, these are selected clones. So these are not ideal weapons that we would like to have. But they work in clinics, they work for many patients, they save lives, and that's a beautiful story. Still, in the future, or we hope this future is going to come fast, we want to move to the right part of this picture. Yeah, we want to be antigen specific in correcting these mistakes made by adaptive immunity. And how much time do I have? Just 10 minutes. Oh, it's okay. So maybe I'm speaking even too fast. Okay, then it's okay. So then, yeah, stated all this, that was the concept, and some things around. And now I'm going to jump to one particular story, and there are many time I will devote to this. And one of the things that we could do more or less antigen or clonally specifically is to specifically suppress or eliminate autoimmune clones. Yeah, that's what we would dream to do instead of using some anti-TNF-alpha or anti-IL-17, while anti-IL-17 is a beautiful drug. But still, it helps only for a portion of patients. What we would like to have is to suppress autoimmunity antigen specifically, and this theoretically should allow us to cure many diseases. And here I will just repeat the question Karnita just asked. So could we ask the disease-associated TCR motives? Because to eliminate something, you need to recognize it first, right? And this is a very, very challenging task, actually. We simplified it for ourselves. Actually, it was a random choice that we had chosen this particular disease. We focused on HLA-B27 associated autoimmunities. It's ankylosing spondylitis, some portion of reactive psoriatic arthritis, and some portion of Crohn's disease, and some others. And it is all linked to one particular HLA. And it simplified the task for us initially because we hoped, and we are more or less probably right, that we are hunting for some particular antigen presented in this HLA-B27, which we hoped could be recognized by a group of similar T-cell receptors. That was the initial hope and idea. And it happened simultaneously. Okay, sorry for this shift here. So adaptive biotechnologists identified these motifs just on a large cohort of patients. They just had a lot of patients. It helps, really. And we did it with a more narrow cohort of patients, but with some nice, beautiful bioinformatics developed by Michael Pogorelli. He is now in Paul Thomas' lab and continuing doing beautiful things there. So what is shown here, that's the number of donors. We have identified some TCR motifs. Or particular TCRs in this case. And here is the probability of rearrangement, which you can really model, and this works quite efficiently. And so the higher the probability that the T-cell receptor be produced in thymus, in general, at average, the higher number of donors you observe in it. But there are outliers, yeah, they behave strange. They are not that probable, but for some reasons you meet them in high proportion of donors, and you look, and you see that these donors were actually HLA-B27 healthy and in disease. And these are all about disease patients, and it is all about the same single motif, very clear one, and it is all about TRBV9, the gene segment inside, which is shared by many, if not most, patients with ankylosing spondylitis, probably by most patients, as we understand now. So we did it like simultaneously with adaptive, and it was in like 2017. But I must state here that the motif itself has been known long ago before, because the very same motif has been actually published in Tissue Antigens in 2002. Yeah? And it was associated there with reactive arthritis. It is like autoimmune-like condition, which is actually the infectious triggered condition. When you observe these clones like operating quite prominently in the repertoire, they grow to the top frequencies. And so the question is, why did it take so long? Because, okay, we could find it much earlier because we should have been actually understanding what we are looking for. And I would answer by a citation of Sidney Brenner, who said the beautiful thing, that the progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So we need to develop techniques, with civilization. Yeah, we need to develop next generation sequencing. We need to develop immune repertoire profiling techniques. Adaptive started in 2008 with DNA multiplexes. Oh, sorry. We started in 2009 with template switch from RNA. And we, like in parallel, developed it. And then we get the opportunity, and the software, MyXCR, was developed. We get opportunity to actually identify these clones because there's autoimmune clones in the blood of patients. It is like 1 per 10,000 T cells. In reactive arthritis, it goes to the top. It can occupy like 1% of repertoire. It's easy to catch, yeah? When it is that small, you need really reliable technique to find it. And then you can try to make a discovery, not the vice versa. So then closer to the pathways with Christophe Garcia, we did some screening, de-screening, and identified epitopes, the bacterial one, the self ones, and so now we can say that the whole consequence of events is more or less clear in molecular level. So again, the concept has existed for many years, and our very good or best friends Andy Sewell published this manuscript in 2012, and we suggested that it should be like this, but nobody never observed it, yeah? So the concept here, is that there is some initially pathogen-derived peptide, okay, bacterial reactive arthritis, which can convert to the bacterial disease later on. This T cell receptor, maybe with lower affinity, recognizes that self-peptide and starts attacking our own tissues because this effector T cell made wrong decision, remembered, overcame the Treg activity and became the stubborn self-attacking clone. That was the concept. But now the difference is that we know it. We know the T cell receptor, we know this antigen, or several of those, and we know this antigen, or several of those. Almost. Okay. There are some hesitations, but it looks like we know the whole consequence of steps right away. And we know the target, importantly. We know the T cell receptor we want to attack. And so if you have the target, you now can think about what you can do to eliminate it. And we developed, together with this pharma company, the antibody which specifically recognizes the TRBV9 segment, which is actually in like 4% of all our T cells, not only in autoimmune clones, of course. So it's not yet ideal drug. Ideally, I would like to kill those 0.01% of cells, and only those. We don't yet have such a drug. And it's a big challenge to develop, but it is already very, very nice story. And it is not suppressing any branch of adaptive immunity. So it works like this. We have different cells. In this case, the autoimmune ones are among CD8, and okay, TRBV9 would be the orange ones. And I know that among orange ones, there are autoimmune clones. And when I add the drug, I kill only those orange cells. Yes, I kill a little bit of everything, but I'm not systemically suppressing neither Treg, not H1, not H2, nobody. So there is no immunosuppressive consequences. And the repertoire is overlapping the many clones, competing as we discussed. So it looks like, knock the wood, it is not a problem for immune system at all. Okay? And it was confirmed in animals that it works and that it is safe. Eliminates the TRBV9. And after that, and actually it happened in 2019, so it happened quite some years ago, we treated the first patient with ankylosing spondylitis like this. And there was a long history we are not going to talk about, but here's the moment when we started the therapy, and it continues nowadays. The patient gets like 2 or 3 injections per year just to be sure. We are not sure we still have continued, but we do it. So the TRBV9, of course, disappears as it should be. But you see, initially when TRBV9 disappeared, in this period, patient felt super nice. Disease disappeared. He said, okay, I'm healthy. I don't want this drug no more. The clone is killed, so I want to go. And we made a pause of interval 8 months. And in 8 months, the TRBV9 here you see appeared a bit, and what is worse, the autoimmune motif appeared a bit. Here it is appearing. We have techniques like double sensitive sandwich to capture them at very low frequency. And in this particular period, the disease also returned. So this is activity of disease. It goes down after we started the therapy. And nowadays, it is just 0. It is not comparable with how he felt when he was on anti-TNF alpha. Disease really disappeared. And still, oops, sorry. Back. And at the moment when the clone returns is associated with the moment when disease returned, and right after the injection, again, it disappeared. And this is about like body flexibility. It also goes down, and so now he feels much, much better. So yeah, altogether, to summarize the second part, now the safety and efficiency confirmed in the phase 2 clinical trial. Phase 3 trial is running. Drug has got fast track registration. And so patients should be able to do it out of the trial. I don't know, maybe starting from this autumn. Yes, I'm finalizing, just 30 seconds. But what is more important for me is that we just show that it is in principle possible. There are other techniques to treat autoimmunities developing. There are tolerogenic vaccines developed by BioNTech. There are Treg TCR-T therapies. Altogether, we know now that we really can cure the autoimmunity if we know the target, and that's the biggest thing for me, and we are moving with this to the right part of the spectrum, and this is, okay, some part of our team. It's really big and international nowadays. Ian, thank you so much for your attention.
Okay, so in the interest of time, I think we have time for maybe one question at most. So from the Slido, let's put that up. So in light of TRBV9, in your opinion, do we need to revisit the idea of definition for all public and private clones?
No, we shouldn't. Let's use another question.
Okay, let's use another question. Have you considered a bispecific antibody for TRBV9 deletion?
You mean, rather we're thinking about beta and alpha to make it more selective, because we know the alpha chain as well. That's the answer.
Okay, all right. I think we'll cut it off there because we have to stay on a very strict timetable, but thank you so much. Okay.